Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 4th February 2021. As we are into the revision mode, we are revising the entire syllabus in the form of sample papers being answered. And in this session we are dealing with sample paper 1. And in this sample paper, yesterday's session, we have answered section A, which is very important. And now we are going for another important section, section B, which is 2 marks. So for your batch, as a new format, there will be 9 questions in section B. So 9 into 2, 18 marks. Yesterday we answered section A, which is 22 marks. Now we are answering section B, which is 18 marks. 22 plus 18 out of 70, 40 marks. If you are thorough with 2, section A, section B, 40 marks will be the available to you. So here we go. Section 17th question. What way the advent of the birth control pill have resulted in an increase in STDs? Answer. The advent of the birth control pill makes it unnecessary to use other means of contraception, particularly condom. Thus, STDs are more readily transmitted during sexual activity. So this is the negative effect of birth control pill. Question 18. In the following pedigree chart, state if the trait is autosomal dominant or autosomal recessive or sex-linked inheritance. Give reason. So there is a pedigree chart. Okay, 3 generations are given, and some are having square blacks, some are round blacks. And in square ones, some are dotted, some are open. Similarly, round ones also, some are dotted, some are open. So based on the explanation that I had given to you in our normal video classes time, you need to understand that it is autosomal dominant. Autosomal dominant. The defective trait is present in both, now look at this, male and female progeny. As you are seeing in F1 generation, both male and female has got the disease, and the unaffected child did not pass the trait, as you are seeing in the diagram. So it becomes autosomal dominant. Question number 19. Tobacco plants are damaged severely when infested with Meledizine incognitia. Name and explain the strategy that is adapted to stop this infestation. Now the answer. The strategy is based on the process of RNA interference. It involves blocking of specific mRNA due to complementary double-stranded RNA molecule that binds to and prevent translation of the mRNA. Hence it is called silencing of mRNA. So this is the best technique in which we can stop this. That is, by silencing of mRNA, we can block it. Question number 20. How does dsRNA, double-stranded RNA, gain entry into eukaryotic cell to cause RNA interference? Now the answer. dsRNA gains entry into eukaryotic cell either through infection by virus having an RNA genome, second, mobile genetic elements, they are called transposons, that replicate via RNA intermediate. These are the 2 ways that DS can enter. You have got a choice for this. Question number 20. What is the choice question? Why are E's used extensively for functional expression of eukaryotic genes? Why are E's used extensively for functional expression of eukaryotic genes? Now what is the answer? E's are the simplest single-celled eukaryotic organisms like bacteria. They reproduce asexually by budding or fission, genetically well characterized, easy to grow and manipulate. They can be grown readily in both small culture vessels as well as large culture bioreactors. Okay. Now question number 21. Name the category of virus that carries reverse transcriptase. So So, transcription and reverse transcription. What is the purpose of this enzyme? Retroviruses carry reverse transcriptase. This enzyme catalyzes the formation of DNA from RNA, which integrates with DNA of the host cell. Normally, what is transcription? DNA to RNA, no? Now, reverse transcription from RNA to DNA, formation of DNA from RNA. Question 22. Give the technical terms for the following. Remember again, in our normal video tutorials time, I told you for every chapter you must remember two things. One is the technical words, the second one is the abbreviations. So there will be definitely questions based on expand the abbreviation as well as technical words. Now, question 22. Give the technical terms for the following. 1. Molecular scissors. Second, molecular sieve. Third, molecular glue. Fourth, autonomous replicating circular DNA. Fifth, first isolated restriction endonuclease. And sixth, extraction of DNA fragments from gel. So first one, all of us know, molecular scissors are always restriction endonucleases. And second one, of course, molecular sieve, agar gel. And molecular glue that attaches means you must remember ligase, DNA ligase. Fourth, autonomous replicating circular DNA, of course, plasmid. And fifth, first isolated restriction, all of us know, Hind2. And then last, extraction of DNA fragments from gel, elution. So this is how you need to remember. And you got a choice in case you are not able to answer these technical words. You got a descriptive type 1. That is, explain the importance of selectable marker with the help of a suitable diagram. Now, when we say help of suitable diagram, first to draw the diagram. Look at this. So you need to draw this. This is PBR 322, very famous, familiar one. And you need to add labeling, that is ORI, don't forget, origin of replication, ampicillin resistance, tetracycline resistance, ROP, and then PVU, SOL1, BAM H1, PST1, PVU1, ECO-R1, CLA1 and HIND3. So these are the various labels that you need to do it. Then you write, selectable markers help in identifying and eliminating non-transformants and selectively permitting the growth of transformants. Normally, the genes encoding for antibiotic resistance to antibiotics such as ampicillin, that's why we said ampicillin R, tetracycline, TET-R, kanamycin or chloramphenicol, etc. are considered as useful selectable markers for E. coli. So here you can see the E. coli cloning vector PBR3 showing antibiotic resistance genes and AMPR and TET-R. Question 23. What is red list? Give two uses of red list. Now, IUCN, International Union for Conservation of Nature and Natural Resources, maintains a red data book or red list, which is a catalog of taxa facing risk of extinction, endangered ones. It aims to convey the urgency of conserving threatened species to the public and policy makers so as to reduce species extinction. Uses: identification and documentation of endangered species of plants and animals, creating awareness about importance of preserving the threatened biodiversity. This is important. Question 24. What is biome? Name the major biomes of India. A biome is a complex of communities with specific types of flora and fauna and maintained under distinctive climatic conditions, that is desert biome. Major biomes of India are tropical rainforests, deciduous forests, deserts and seacoast. Now question 25. How do scientists extrapolate the total number of species on Earth? Answer. Considering that an overwhelmingly large proportion of the species waiting to be discovered are in the tropics, biologists make a statistical comparison of the temperate tropical species richness of an exhaustively studied group of insects and extrapolate this ratio to other groups of animals and plants to come up with gross estimate of the total number of species on the earth. That means we are taking a sample survey. Based on that sample survey, we are calculating the actual numbers. Now we move on to section C, that is 3-mark questions. And as you know, there will be 5 3-mark questions. 5 into 3, 15. So here we go with at least one question today in this session so that we can cover others in the next session. Define point mutation. Give one example. Mutations are new, sudden, inheritable, discontinuous variations. So whenever we say mutation, it is sudden, all of a sudden it takes place. And this appear in the organisms due to permanent change in their genotype. Point mutations are a type of gene mutations which involve the substitution, deletion or insertion of a single nucleotide. Remember, when we talk of the nucleotide or nitrogen base, like we said, adenine, guanine, cytosine, uracil. So one nitrogen base is missing, then we say it is point mutation. So point mutation is strong. Example, sickle cell anemia. An autosomal hereditary disorder is caused by the formation of abnormal hemoglobin called HbS. Differs from normal hemoglobin A in only one amino acid, that is 6th amino acid of, look at that, this is important, only one amino acid, 6th amino acid of beta chain of glutamic acid is replaced by valine, just one amino acid. Thus, disorder is due to change of one nitrogen base in cistron. Okay. Now, in our next session, we will answer remaining 4 questions of 3 marks and 5 marks questions of 3. So that keep a note separately, and all the 10, I am going to give you 10 sample papers. Write